significance is accurate or not. So now, if we look at the box whisker plot, for number of hemorrhages by group treatment, what do we see? We can see that there is significance between the number of hemorrhages between all three groups. We see that in the control group that has no treatment of angiotensin 100, there is a higher number of hemorrhages versus either of the angiotensin 2 groups. The reason why we have an asterisk there is because if you look at the legend, and I know it's hard to see. Yeah, I'm not going to be able to show that. But if we look at the legend, pretty much what the asterisk signifies is the level of significance that there is between the groups. So if you have an asterisk of 1, then that signifies you have a significance that's below 0.5 but greater than 0.1. And if you have 2 asterisks, that will show that you have a significance that's below 0.01 but greater than 0.0001. In this case, each group has 1 asterisk because their significance is below 0.005. If we look at the length of hemorrhages by group treatment, we actually saw that there was no significance between the average length of the hemorrhages, which tells us that infusion of angiotensin 100 and 500 may not have an effect on the length of the hemorrhages. And then last but not least, these are some of the stains that we got using the hematoxylin and eosin staining. So pretty much what you can see here is you see 3 photos. This first photo is a photo of the control group. And you can see that when you compare it to both the other photos, the second one being a photo of the angiotensin 100 group and the third photo being a photo of the angiotensin 500 group, you see that there are far more number of hemorrhages prevalent in the control group, which is consistent with the graphs that we have here, because pretty much what that tells us is that we do have higher numbers of hemorrhages by group treatment for the control group as compared to the other 2 groups. And we also have higher levels of parasitemia, basically higher levels of parasites in the blood for the control group compared to the other 2 groups. We also have a lower level of survival probability for the control group, while we have higher levels of probability, survival probability for the angiotensin 100 and 500 group.
I'm just giving you a quick reminder that you are getting close to time.
Thank you. I'm gonna present this on the bigger screen. So to conclude, earlier studies on the effect of angiotensin 2 and cerebral malaria have shown that angiotensin 2 can protect against the development of cerebral malaria, because when we looked at the data, as I mentioned before, we saw that you had lower levels of parasitemia in the blood, you had a higher probability of living longer, and you had fewer numbers of hemorrhages for both groups that were treated with angiotensin 100 and 500 when compared to the control group. In the study, we basically wanted to explore these effects, and again, the data pretty much answered or showed that our hypothesis that if you use angiotensin to treat cerebral malaria, you can impact it by pretty much inhibiting or slowing its effects. And just wanted to acknowledge Joseph Racklin and Julio Gallego Delgado. Joseph Racklin, pretty much said what his occupation is. He's the director for the Laboratory for Marine and Estuarine Research. And Julio Delgado served as my PI. And he also teaches at Lehman University. And that's the end of my presentation.
Thank you so much, Ezekiel. I mean, thank you so much, Mousa. That's fantastic. And really wonderfully presented and really wonderful posters. Who has questions?
Yeah, I'm trying to exit out of the presentation so I could check the chat to see if there's any questions.
Yeah. You have some like that was great chat, but if anybody has a question, just unmute your microphone or put it into chat. Mouse is getting a lot of love in the chat, which is lovely.
Oh, thank you, everyone. I appreciate it.
It was really interesting. I have to tell you, I'm learning so much today from all of you. You're amazing.
Thank you. Thank you.
If we don't have any questions, then we're going to get ready for Ezekiel, who's been waiting in the wings. And I want to give 2 points. If maybe what would make sense to sort of facilitate having questions when the person is done presenting is if you're listening, like as you're listening, maybe you can write down little questions in a notes app or on a piece of paper as you're listening, or you can just put the questions in the chat, right? The presenter will do their presentation, and then when they're done, they'll look over at the chat and answer your questions. It's a ton of information coming at everybody, and sometimes our questions kind of emerge at particular points. And if you want, one way to make sure that the person's getting some nice feedback is if you write your questions out in the chat as they come to you as you're listening. And then the other important point is that we want to keep the presentations to 10 minutes, which is tough because you're clearly doing some pretty advanced and deep research. But check the clock. So with that, I'm gonna welcome Ezekiel. And it's now 1:35. So at 1:45, we want to be asking him questions. And welcome, and we look forward to seeing you, Ezekiel.
Thank you. Can you guys hear me?
Yes, we can hear you, and we're going to just continue recording since you gave permission to have your presentation recorded. So thank you. We're going to keep that up.
Yes. Thank you, guys. So today, my name is Ezekiel Olumoide. I'm a junior, and I'm a biochemistry major. I'm working under Moira Suwani, and today I'll be presenting mechanistic aspect of interleukin 24. So I'm just going to zoom in. Yes. So cancer cells, what makes cancer cells different from normal body cells is that they overexpress some genes to produce, to upregulate some survival and proliferation proteins that makes them grow continuously, and they lose restraint over the translational machinery, which is the mRNA to protein translation. So what we are investigating is a protein called interleukin 24, which is also called melanoma differentiation associated protein, and this protein is known to cause different apoptotic effects on cancer cells, and an overexpression of this protein could not only inhibit the metastasis of apoptosis of cancer cells, but also inhibit other functions of cancer cells. And what is great about this protein is that it does not affect normal body cells. So that was one of the great things about this protein. So what we do know about cancer cells is that cancer cells are different in every human body and in every individual, and even in the same individual, it could be different in the sites, in the microenvironment sites. So, but what we wanted to know is that what makes them common, and we do know that what makes them common is that translational initiation, that all of them have to go through translation to create that protein. So what I wanted to know, our purpose of the study is to know if interleukin 24 affects the translation of some of these proteins that, or oncoproteins that cancer cells use. So meaning like if interleukin 24 inhibits like a initiation factor, EI4A, then it will produce less oncoproteins, and then that would lead to apoptosis and cell decrease, proliferation, and cell death. So what we did, our experimental methods were viral infection. Then we took a non-replicating virus, adenovirus, and which contains interleukin 24 gene, and we infect it into the cancer cells. And this virus then releases the interleukin 24 gene, which gets transcribed and then get translated and also secreted. And this secreted gene can bind to the cell, either even in the ER of the cell or on a receptor on top of the cell, and also binds to other cancer cells and causes apoptosis. And to do this, we use cell culture. This is ELA cells, and this is MCF7. This is ELA cells are cervical cancer, and MCF7 are breast cancer cells. So we also use KRT-PCR, we use Western blots, we use Luciferase Assay, and Eosin V binding assay. So our results, what we got was interleukin 24 inhibit the gene expression of 5' untranslated region. So this is the so what we do know is that Ocon proteins in the mRNA, they have some secondary structure on the 5', the upstream of the the mRNA. So then we did Luciferase assay to look at different mRNA structures and look at the effect of interleukin 24 on that translation. So looking at this, you could see that there's no difference between all these structures that don't have this loop in the mRNA. So you could see that there's no difference, but in this, there's a difference, which shows that interleukin 24 actually affects the cleavage of this loop. So we know that EI4A is a helicase, is like a scissors that cuts this loop into two to straighten it out for the other translational initiation factors bind to. So we treated interleukin 24 to different cancer cells and saw that it causes downregulation in all cancer cells of EI4A complex, which correlates with this data. So we also checked for, sorry. Okay. So we also checked for the if interleukin 24 induced apoptosis through this, and we saw that the black means interleukin 24, the white means the control group. So we compared and we saw that interleukin 24 increased cell death in all these different cancer cells from different sites, from melanoma, from prostate cancer, from breast cancer, and from cervical cancer. And it also increased the apoptosis of all these different cancer cell types. And we also showed that interleukin 24 causes the upregulation of caspase-3. Caspase-3 is a pro-apoptotic protein that cleaves different proteins, that degrades different structural proteins in cells, not just cancer cells, but also normal cells. So since we know that interleukin 24 decrease EI4A protein, then we decide to treat the cells with EI4A and see if there will still be, if there's going to be apoptosis or cell death. Then we did that and saw that EI4A rescues the apoptosis and cell death of interleukin 24. So that shows that for interleukin 24 to cause its killing effects, we need EI4A downregulation. This here. Sorry. Close this, yes. So now we decide to look at different oncoproteins, CDC25C, CMYC, ODC, and X-linked apoptotic protein, X-linked inhibitor apoptotic protein. Decided to look at the protein level, and we saw that treating them with interleukin 24 downregulates all these proteins. And all these proteins have, in the mRNA, they have the secondary structure, this secondary structure. So it does correlate with our data that interleukin 24 downregulate, interleukin 24 mechanism of killing is mediated through EI4A downregulation. Then we decided to, since we saw that it decreased the protein level, we decided to look at the mRNA level of all these oncoproteins, and we saw that there's no difference in the mRNA level of all these oncoproteins. Then we concluded that interleukin 24 inhibits all oncoproteins through the translational level and not the transcriptional level. Then previously, we showed that interleukin 24 physically binds to sigma-1 receptor. Sigma-1 receptor is a chaperone protein that is useful for folding of cells and also secretion of most oncoproteins, and also checkpoint inhibitors like PDL1. So we showed that, partially we showed that sigma-1 receptor binds to interleukin-24, and interleukin-24 have an antagonistic effect on sigma-1 receptor function. Then we decided to see if sigma-1 receptor rescues interleukin-24 inhibition of EI4E if it is upregulated. Then we decided to compare. Then we use EI4 sigma-1 receptor agonist, which is SKF10047, which causes the increase of the function of sigma-1. And we saw that when we treat this cell with the agonist, the sigma-1 receptor agonist, there's no difference. There's no difference between it and sigma-1. I also showed that sigma-1 is actually very important for EI4A, for interleukin-24 inhibition of EI4A.
Ezekiel, I'm going to have to ask you. Oh, here you are. The conclusion slide. Perfect.
Yes, I'm on the conclusion. So we concluded that EI4A inhibits translation of EI4A, and also it does this through sigma-1 receptor. Then our future research is to look at how does it downregulate EI4A. Then we postulated these 4 apoptosis that is through prostaglandin, through a tumor suppressor, through miRNA, and through a champion-mediated autophagy. Then I would like to acknowledge my lab mates, Christian, Justina, Ilal, Gabriel, for the feedback. And also I would like to thank the Lehman College Department of Biological Science for the fellowship award, for the 2020 fellowship award. I also like to thank the National Cancer Institute for the grants that helped with this project.
Thank you, Ezekiel. That is such important work, and you are doing, potentially life-saving work to help people who are suffering with cancer. So we appreciate all the work that you're doing, and you should be very proud of yourself.
Thank you.
Yeah, that is. And if you look to the chats, you'll see some questions there.
Yes, I saw a question right now. Is it advantageous to inhibit oncoproteins in during translation? Yes, it is, because oncoproteins are generated from oncogenes. Oncogenes are mutated genes that causes cancer cell growth. So by inhibiting these oncoproteins, you are reducing the survival of all these cancer cells, their proliferation, and you are causing apoptosis. So it's a really good therapeutic method by just inhibition of oncoproteins.
And do we have any other questions? Really excellent job, Ezekiel. Thank you.
Thank you, Ezekiel. Nice to see you.
Excellent. And so next up we have Kelsey Goon, whose paper is Not Just the Tibia Sidekick, Measuring the Relative Cortical Area, Osteon Area and Shape, and the Osteon Density of the Fibula. So Kelsey, you've said that you are, you consent to being recorded. So we're just going to keep the recording going if that's all right.
Yes, that's fine. Thank you.
Very nice. All right. Go ahead.
Just going to share screen here.
Mm-hmm.
Okay. Let me go to my, of course. All right. Might take a second. Okay. Hello, everyone. My name is Kelsey Gounet. I am a biology student here at Lehman, undergraduate, previously an anthropology, biology, chemistry student as well. So now I'm minoring in anthropology. So the purpose of my research today is going to be using histological measurements in order to understand the relations that the fibula can be used for in the field of forensic anthropology. So I'd like to start by explaining some of the methods and why forensic anthropologists use these methods. So forensic anthropology uses histological methods, which is basically methods looking at the microstructure of tissue to determine things like age, sex, and stature. And oftentimes skeletal remains are used for this purpose. One of the ways to determine age using skeletal remains is by identifying signs of bone remodeling. Now, bone remodeling is the way in which our bones maintain and repair. It's a system of maintenance and repair. And in order to see a little bit of that, I'm going to direct you to figure 2B. You can kind of see it here. Here we have a drawing of a zoom-in image of our osteons in our fibula sample. So here you can see a Haversian canal, which is the empty space here. It's an empty vascular space. And here we can also see like a tree ring pattern. So I'm going to explain a little bit of that as well. So as we age, osteons accumulate. So because of this accumulation, we are able to use it in age determination using skeletal remains. So similar to a tree building rings, our bodies also build rings in the form of osteons from bone remodeling. So oftentimes in this field, the femur or the rib is used, and this is because it's very well researched and because it's also very accessible anatomically. If you think about the femur, for example, that is your kind of like, I guess I should just call it the thigh bone for those of us that aren't, bone histologists. And then the rib, of course, is in your rib cage area. So why do we want to use the fibula is a good question. We want to use the fibula because it's something that we can use in addition to the rib and the femur. The fibula, unlike the rib and femur, doesn't have as much gross morphological use. And by that, I mean, as a whole, it's not as useful for things to identify different characteristics of the body. So because of this, it's a little more disposable. So it might be a nice addition. And we have seen in previous research in 1965 by Dr. Curley that has asserted that the fibula may be the most accurate bone to determine age. So essentially, my objective in this experiment is to figure out whether the fibula is a viable addition to the rib and femur in terms of measuring things like relative cortical area, osteon area and shape, and the osteon density of the fibula. All right, and to do that, we would like to identify trends relating to age and age determination. So in terms of how we conducted this experiment, I used an image software called ImageJ along with a cortical area macro set in order to find the cortical area of each fibula sample. Now, our sample is 5 individuals ranging from age 44 to 96, and they are all females. So this means that all of our samples are in the postmenopausal phase, and that's going to come in a little bit later. So in terms of measuring our intact osteons, so one of the things we measured was the density of intact osteons, also known as OPDI. And I'm just going to zoom in here again. So you can see here our osteons are circled in these red lines. So the reversal line here on our osteon is going to be that edge that's circled here. So we're essentially manually tracing the reversal lines of each osteon to get the measurement of its area and shape. In terms of the cortical area, we simply, you can zoom in on this image, we simply traced along the inside of the bone, or I guess the bone marrow cavity, most people know it as, and the outside of the bone, periosteal bone, and did a simple subtraction to find the area of the actual cortical bone. In order to set up a criteria to trace the osteon, so you can see there's not much, there's a lot of different shapes present in these pictures. I had to set up a criteria so that it was very standard. So one of the standards was that it was 50% or more of the osteon had to be present in each of these grid squares. The reversal line must be present and visible, and that the Haversian canal had to also be present, Haversian canal being this empty space. And in order to basically make our research both easily understandable and quick, considering the crisis that we're in right now, we decided to use 50% of the fibula cross section. And to do that, we used a grid pattern, a grid with a checkerboard pattern to obtain 50% of the cross sections. And these are all cross sections from the midsection of the fibula. So here you can see the grid pattern we created and a zoom in of one of our grids squares, which is from B6. You can see the numbers in it too. So as a result of all of our collected data, we found some significant trends that matched with the previous research done on these topics. The main one we saw that was a significant trend was between age and cortical area. Just to remind you guys, this one. So the cortical area did decrease with age, which was to be expected. However, the age and osteon area and the age and circularity, which is from a scale of 0 to 1, 1 being the most circular, 0 being the most abnormally shaped, there wasn't a strong correlation, which is also to be expected based on previous research. The age versus OPDI, or osteon population density of intact osteons, was an increase with age, which was also to be expected. And we can see that here in figures 4A and B, as well as figure 3 here. So I would like to get into my conclusions. So our experimental values did follow the trends from previous research. However, we do need to keep in mind that we had 5 samples, which is a rather small sample. Therefore, our study, there needs to be a little bit more study into this topic to confirm. Our cortical areas did decrease with age, which was consistent with research that had previously been done. And typically, in these types of studies, they would use the OPD, or just osteon population density. But due to our lack of ability to use the microscope to check the images of the slides, it was a little more advantageous to use the OPDI. And regardless of that, we did see confirmation and trends, or a correlation, regardless of the fact that we didn't include things like osteon fragments or drifting osteons, which would be included in the osteon population density. So in addition, I would like to acknowledge the donors of our slides. And I would also like to thank Dr. Amanda Agnew and Dr. Sam Stout for the slides that we were able to use that they created. Thank you.
Thank you so much, Kelsey. That's fantastic. Does anyone have any questions? I'll leave that for a minute if people want to gather their thoughts and ask a question on Kelsey's research. But thank you very much, and thank you for keeping to time. We're going to be queuing up Crystal Joinville, who asks actually not to be recorded. So we're going to pause this recording now so that Crystal can give her presentation. But if there's any last questions. No. All right. So then, Crystal, welcome. We're excited to have you. Crystal's presentation is bioaccessibility of cadmium and zinc in wheat seed. And I'm going to stop recording now. And the reminder is just please keep it to 10 minutes, and so that we'll be finishing at 1:10. I'll give you, I might break in at 1:09 or send you a chat at 1:09 to say, okay, wrap it up. And then the other reminder is to everybody who's listening, if you have questions as the presentation is going, just like you would if you were taking notes, put them in chat, right? And then you'll have questions when we finish at 1:10, I mean at 2:10, you'll be able to say, here's my question right away. So if you have questions as Crystal's giving her presentation, why don't you put them in chat, and then we'll be able to speak them at 2:10. And so here we go. Thank you.